Welcome to Baking with Biologists. Today we will be discussing the phases of meiosis with the help of cupcakes. This is a cell in the beginning of interphase. The DNA is in its least condensed state, chromatin, jumbled together within the nucleus of the cell. Half of the genetic information is inherited from the mother and half from the father. To make it easier for demonstration purposes, we have color-coded the chromosomes inherited from the mother red and the chromosomes inherited from the father blue.
Throughout interphase, this cell essentially prepares itself for meiosis, growing in size and synthesizing the proteins necessary for this task. These cupcakes are a zoomed-in look at what is going on inside the nucleus during S phase. The chromosomes that are the same size are homologous, meaning that they contain the same genes, but may have different alleles. During S phase, exact copies of each chromosome are created to form a pair of sister chromatids. These sister chromatids are identical to one another and are attached at an area called the centromere, held together by cohesin proteins.
In prophase 1, the cell remains diploid, meaning it contains two complete sets of chromosomes, one set from each parent. In this representation, the cell has 2n equals 6 chromosomes, indicating that there are two copies of each of the three types of chromosomes to create 6 total chromosomes. At this point, the chromatin condenses into individual chromosomes. During this time, the centrosomes begin to form spindles. Synapsis occurs in prophase 1, in which homologous pairs of sister chromatids wrap very tightly together. Because of this, crossing over occurs, creating new chromosomes as genes on chromosomes from one parent switch with genes on chromosomes from another parent, shown by the blue chromosomes having some red and vice versa. Additionally, the nuclear membrane starts to break down as shown by the dashed purple line.
In metaphase 1, the homologous pairs of chromosomes create a line down the center of a cell, along what is called the metaphase plate. This relates back to Mendelian genetics, specifically the law of independent assortment, because independent orientation of the chromosomes occurs, meaning there are multiple orders the chromosomes can situate themselves on the metaphase plate. The traits inherited by the offspring are therefore independently assorted because the way one pair of homologous chromosomes orders itself has no effect on the way another pair does. At this point, the centrosomes of the cell are located on the opposite poles of the cell and their spindles attached to the proteins on the centromeres of each chromosome. The homologous chromosomes are each attached to the microtubules coming from opposing poles of the cell. In anaphase 1, the pair of homologous chromosomes separate, each chromosome being guided via a spindle apparatus to a different side of the cell. The sister chromatids remain attached at the centromere.
In telophase 1, two new haploid nuclei form within the cell, meaning each nucleus contains only one set of chromosomes. Simultaneously, cytokinesis begins to occur, during which the single cell is divided into two new daughter cells, shown by the two cupcakes nearing complete separation. These are the two new daughter cells.
In prophase 2, the newly formed nuclear membrane in each cell breaks down again.
The centrosomes move apart during spindle formation.
In metaphase 2, the chromosomes line up along the metaphase plate, and the spindles attach to the proteins at the centromeres of the chromosomes.
In anaphase 2, each pair of sister chromatids is separated by a breakdown of the protein that held them together. Each sister chromatid, now individual chromosomes, are guided by the spindles to the opposite sides of the cell.
In telophase 2, nuclear membranes reform around each set of chromatids located at the poles of the cell. During cytokinesis, each cell divides into two new cells, creating 4 haploid daughter cells that are genetically different from one another. In humans, the only haploid cells we have are gametes, or sex cells. When a sperm cell fertilizes an egg cell, the zygote created is diploid because it contains a set of chromosomes from each parent. As a human grows and reaches sexual maturity, the somatic cells, or body cells, remain diploid, but the gametes produced during meiosis are haploid, and during reproduction the haploid gamete cells meet, thus starting the cycle all over again. This concludes Baking with Biologists. Hope you enjoyed!